The cells of bacteria, fungi, plants have thick but porous coat outside their inner structure, and that is known as the cell wall. Inside the cell wall lies the plasma membrane. The composition of the cell wall varies for different groups, like it is different for the plants, for fungi, for even a bacteria. So it varies for each and every organism. But there is some similarity in the kind of substances which are used for making the cell wall. The purpose of the cell wall is to protect the inner plasma membrane because of the hostile climatic conditions which the organism, like the fungi or the bacteria or even the plants, they have to withstand. And as because they are not having the outer covering like the skin or outer defense mechanism, immunity, these all things are lacking in most of the um, organisms which have the presence of cell wall, and that's why they have presence of an outer tensile or hard covering. Now this outer covering, it is made up of, it has certain chemical composition. The chemical composition in case of various organisms is of various types, but we are going to discuss about some of the basic chemicals which are used for formation of the cell wall for different organisms. Cellulose is a molecule which is a long unbranched chain of glucose, and it is used to make microfibrils. Like you can think of making forming certain small fibers, and these fibers they all combine together to form microfibrils, which ultimately forms the structure forms a structure which is this kind of structure, the meshwork you can see. This entire meshwork has cellulose presence of cellulose, that purple color lining, that is actually the micro or macrofibrils which actually lead towards giving the entire structure of a cell wall. It forms the tensile outer covering. So that is something which is very important, and it is also known as a ground substance because the microfibrils are the ones which act as steel. So if you think about an entire structure or a building which is going to be constructed, we need the iron rods to be present at the corners to give support. The That support is being provided by the microfibrils. They are making the ground substance or the matrix of the plant cell wall. The matrix is itself filled with amorphous jelly-like material. When I say jelly-like material, it means you can think of the outer covering or this outer covering, or you can also think of the inside layer, the white structure, whatever is there, or white, anything which is white visible, that is all made up of substances which ultimately leads towards or that has to be filled with some substance, and it is filled with this amorphous jelly like material which is a polysaccharide and has and is commonly known as the pectins. Here the jelly like substance is made up of not only the pectins or some of the hemicelluloses, it is also containing a high amount of water and some proteins which are all mixed together in order to form a selectively permeable membrane kind of inside content which is filled in this all structure. If you see the entire structure, this entire box which is visible has to be filled with some substance, and that substance is the mixture. That substance is known as the matrix in simple terms. Now what is hemicellulose? As I have mentioned, it is also a kind of polysaccharide, and it is used to form the matrix of the cell wall. Pectins are another kind of branched polysaccharides, and they have the capability of holding water within themselves, and as a result they form a jelly like substance which is fibrous, and fills up the entire space of the plant cell, and because of that the pathogenic attack cannot take place in case of plant cell. And as a result we can think of pectin to be acting as a defensive substance which is present in the cell wall. And as because it is jelly like or gum like substance, so because of that it is capable of trapping or not allowing the movement of any germs inside the plant. There are various proteins also which intervene or which form the entire structure of the plant cell wall. And these substances are, why are we saying them to be proteins? Because proteins are the only ones which are capable of forming secondary, tertiary, and quaternary structures. It means the twist can be formed in the structure of any cell wall or any organism only because of proteins, because proteins are capable of forming multiple chains, and these all chains can further intervene within themselves to form quaternary chains. Quaternary, primary, secondary, tertiary, and quaternary chains can be formed in case of proteins, and that's why they are also required for the purpose of cell wall formation. This is regarding the chemical composition of the plant cell wall. Now, plant cell wall is containing the cellulose, the hemicellulose, the pectins, the proteins, and definitely water content will be there, which is around 60 to 70%, and it keeps the entire plant hydrated. And not only that, it also provides you are aware that if the plant is in a moist condition, then only it is living, otherwise it will be regarded as a dead plant. So that's why it's very important. Now it's also quite important to know few depositions which take place in the plant. In the plants we see that there are lignin depositions are present in the pits, and in the pits there is continuous deposition of tough glue like substances which are aldehydes. And as because these are present in the cells or in the cell wall of the plants become very hard. And yes, they become very hard. Next is the cutin. If you remember, the leaf upper structure or the upper thin layer of the leaf is made up of fat like substance which is deposited over it, and that is known as cutin. Now this substance is impermeable to water, and the function of cutin is to check evaporation. So this is something which you all are already aware of. And what is its use? It is helping the plants. Suberin deposition is there in the, I think everyone remembers. Yes, it is present in the cork cells. And over there, the suberin deposition, it is helpful in um, hindering the movement of water, because of which symplastic mode of water movement is preferred rather than the apoplastic movement. So that is something which is to be remembered. Next is certain other substances, like think about, this is about the plant structure. Now think about the fungal structure, fungal body. In the fungus, the outer cell wall is made up of chitin. We have already discussed about chitin. So chitin is a substance which is a polysaccharide and is present in the outer wall of the mycelium, which makes the outer tough covering in case of the fungi. So basically all these cell wall outer coverings, they are helping in forming an outer covering and giving the structure to a plant body. In a broader term, it is described over here that the cross linking of glycan is a kind of glycoprotein. Similarly, the entire structure we have kind of discussed, and this is the backbone which is shown over here. All these, the blue color structures, the purple color structures, or even the yellow structures, they are all the microfibrils which are all joined together to form macrofibrils, and finally they are giving the entire structure over which the matrix or the entire cell body will be formed or will be getting its entire shape. And what is the function of the cell wall. That is quite important. It maintains the shape of the cell wall or the cell of a organism. See, the entire framework is made. Now if the outline is made, then just the requirement now is for making the entire inside content. So cell organelles or the plasma membrane is to be made after this. It helps in protecting the plant against any kind of mechanical injury. So whenever there is a chance that any pest attack can take place, during that time this chitin deposition or suberin deposition or cutin, these all are substances which are helpful in fighting against the germs. They are not allowing the germs to enter the body. Even the jelly like matrix, it is, think about a jelly like substance, there is no space for a bacterium to roam up, so that's why they cannot grow inside a plant body. But still there are some pathogens which can enter and they grow, but to more or less extent, or to layer. It helps in fighting against any kind of pathogenic attack. Here K is missing, so you can think of writing a K over here. And it also provides mechanical support against the gravity. Now because of that, it is due to the rigid cell walls that the aerial parts of the plants are able to keep themselves erect and expose themselves to the sunlight. If the plants, they are not deep rooted and they are not very strong, then there will be a very less chance for the leaves to be erected towards the sun for getting the solar radiation. So that's the reason why the attraction or the mechanical support against the gravity is very important in case of plants, and that the plants are getting with the outer covering of cell wall only, because it is the hard outer covering. The cell wall prevents undue expansion of the cell, because every time water is falling in by the process of rainfall or even by absorption, not of absorption, by osmosis, the roots of the plant, they are always doing osmosis, because of which the water is moving inside with the help of contractile vacuoles as well as xylem. Now think about the plants undergoing continuous osmosis. If that was the case and expansion of the cells used to starts occurring, then the cells will burst off because it has very high content of water. But since the cells, they are not undergoing continuous osmosis and they are not bursting off, because the evaporation is taking place. And vessels, if you remember, they are made up of dead cells, and because of these complex tissues which are present in the plants, the proper process of elimination of extra product or extra cell content takes place, or extra water takes place. It also allows the materials to pass in and out of the cell, because cell wall is freely permeable. Although it is freely permeable, but sometimes certain regulatory substances, the resins or some gums or any secondary substances, the tannins, they all are released from the outer covering of the cell wall in case of plants. Many enzymatic activities are also controlled with the help of the cell wall, and there is presence of cutin. We have already discussed cutin and suberin deposits are there, and these check the evaporation process in case of the plants. Now there is growth of the cell wall, which enables the cells to enlarge in size. Continuous cell wall is growing in itself so that new cell formation takes place, because outer covering has also to increase its size, right? The cell wall plays very important role in passage of materials from inside and outside of the cell, because outermost covering is the cell wall, so anything which has to go out of the plant has to pass through it. Now there are various pores which are present in case of the cell wall, and these pores form the plasmodesmata, which link up to form symplastic mode of movement in case of plants. Now symplastic mode of movement is always hindered by the apoplastic mode of movement whenever there is suberin deposition. So that is also to be kept in mind. The cell wall in some cases has a role of defense as well, because it has a presence of spines. If you remember the rose or cactus or most of the plants, the outermost covering is having presence of spines. So it is the cell wall which is forming the spines, and that's why it is having a defense, direct defense role, not only acting as a mechanical support and protecting against any outside attack, but also having a proper control over the defense mechanism by formation of spines. So these are some of the important functions of the cell wall. And what about the animal cells? We have discussed only about the plant cell wall. Animal cells, in case of the animals, we see that there is absence of cell wall. Then what else is present in spite of the cell wall? Animals, they have presence of extracellular matrix, which is made up of glycoproteins and three different types of glycoproteins, basically the collagen fibers, the proteoglycans, and at times the fibronectins. These are three basic kinds of integrins are also there. These all form the entire outer covering, or they make the extracellular matrix or the cell coat of the animal cell. Now, because of presence of these all substances, as because they all are protein based substances, and or I should say glucose protein based substances, so they make the outer cell wall, they form receptors, they form the cytoskeleton. So every work is done with the help of only these substances. Or collagen makes the framework, proteoglycans, it forms the receptors. So any substance or any foreign material, if it is trying to enter the cell wall, it has, or plasma membrane, it needs the presence of cell receptors. So those cell receptors are made with the help of proteoglycans. The function of fibronectins is to form receptor proteins. Some of the receptors, like erythrocytes, they have presence of antigens over it. So those receptors may be present in the case of plasma membrane, wherever they have to get attached to. So here I'm not talking about only the erythrocytes, some other substances as well. Suppose a particular protein is there and it has to get inside a human, inside a particular cell. So there it needs a receptor. As soon as it binds to the receptor, it can move it, otherwise it is not allowed to move it. So in that way it is very important. So each of these substances are important in forming the outer covering of the animal cell wall. The function of the extracellular matrix is to protect the cell. It supports and maintains the form or shape of the cell. It helps in recognition of similar cells, but so cell to cell adherence can be done with the help of ECM, or that is extracellular matrix. Sometimes the coordination between two cells is also maintained because of the ECM, and it also helps in the function of the WBCs, and it helps in identification of the microbes with the help of the receptors which have been formed by the proteoglycans or the fibronectins. It also helps in transmission of information from outside environment to the cell, in to the cell's interior. Suppose um, now a pest attack is about to take place or has already taken place, and as a result there has been release of toxins. Now those toxins will be understood or will be recognized by the receptors present on the cell, receptors formed by the fibronectin and the proteoglycans, and as a result the response from the inside of the cell will come out in form of WBCs or in form of certain proteins which will be digesting the toxins or the bacterium or will be fighting against them. So inner mechanism can act, or inner defense mechanism can get activated by the outer environmental conditions which can be sensed by the receptors made by the fibronectins as well as the proteoglycans. Now the extracellular matrix of erythrocytes also have presence of antigens, and this help in the formation or recognition of blood groups. So this is a basic idea about this outer covering of the animal and plant cell. What about the fungi? Fungi have has presence of chitin, and that's it. So this is all regarding the summarization of the cell wall.